the floor where they drop like bombs. Faceless voices running all the cons. Digital whispers in a binary night. No light, no face, pure sound, no sight. Stories unfold in the dark unscrolled. Echoes of silence, secrets untold. Waves crash loud in this audio cave. No mic, no fame, just the sound we crave. Tune and turn up, let the silence break. Raw, unfiltered, hear the earthquake. No mic needed, just a vibe that's real. Faceless words you can touch and feel. In shadows, voices untamed. No need for spotlight, no claim to fame. Just a beat of life in a stream of thought. Every word a punchline, no need to be taught. Just listen close, no visuals needed. Raw conversation, unprocessed, unseated. Feel the pulse in every sentence said. No distractions, just thoughts in your head. Whispers of the streets, rumbling through. Faceless yet vivid, a world in view. No mic, just souls, connecting in flow. Pure and raw, let the rhythm go.
I'm happy to be here. What would you like to talk about? My time as a monk has greatly influenced my approach to both science and life. The order and simplicity of life in the monastery provided me with the necessary tranquility to carry out my research and contemplate the natural world. I found that the rigors of monastic life, such as fasting and prayer, helped me to focus my mind and cultivate patience, qualities that proved invaluable in the long and demanding process of scientific investigation and experimentation. Growing up on a farm, I had a close relationship with the land, plants and the natural world from a young age. This early exposure instilled in me a fascination with the science of plant breeding. I began to observe patterns and variations in the plants, and I wondered how these traits were passed down from one generation to the next. This curiosity and my observations as a farmer later became the foundation for my scientific investigations in genetics and inheritance. Growing up in a peasant family in Silesia, I faced numerous challenges, both economic and societal. My family struggled financially, and my parents sacrificed greatly to send me to school. Additionally, the social norms and expectations of the time were difficult to navigate. As a young peasant boy, I faced prejudices and limitations based on my social standing. However, these challenges also taught me resilience, resourcefulness, and the value of hard work and dedication, which later proved to be crucial in my scientific career. My time at the University of Olomouc was pivotal in shaping my scientific curiosity and approach. The rigorous academic environment, particularly in the fields of biology and physics, exposed me to a wide range of scientific knowledge and theories. The university provided me with the opportunity to engage in intellectual discussions and debates with professors and peers, fostering my critical thinking skills and encouraging me to question the natural world around me. Additionally, the university's resources and laboratories allowed me to gain practical experience in experimentation, which would later become essential in my genetic research. During my studies in Vienna, I was fortunate to have several influential mentors who shaped my scientific thinking. One of the most significant was Franz Unger, a professor of botany at the Vienna University of Philosophy, who introduced me to the world of plant research and provided me with valuable guidance. Another important mentor was Hermann Bonnet, a renowned physicist, who helped me develop a strong foundation in mathematics and physics, which later proved crucial in my genetic work. The professors at the university, with their diverse expertise, provided me with a broad scientific perspective and knowledge that greatly influenced my research in genetics. I chose pea plants for my experiments on inheritance primarily due to their convenient characteristics. Peas are easy to cultivate and have a short life cycle, allowing for rapid reproduction and observation of generations. Additionally, peas exhibit distinct and easily observable traits such as flower color and pod shape, making it simple to track inheritance patterns. This made them an ideal subject for my research on inheritance, as I could easily control the breeding process and observe the results of my crossings. In summary, the practicality, rapid life cycle, and distinct traits of pea plants made them an excellent choice for my genetic experiments. In deciding on the traits to study, I sought those that were distinct, easily identifiable, and preferably monogenic in nature. That is, they were determined by the effect of a single gene rather than multiple genes or environmental factors. I chose 7 traits: seed shape, round or wrinkled; seed color, yellow or green; pod shape, inflated or constricted; pod color, green or yellow; flower color, white or purple; flower position, axial or terminal; and height, tall or dwarf. These traits represented a range of observable characteristics that I could use to infer patterns of inheritance. In my monastery garden, I cross-pollinated pea plants by collecting pollen from the male flower of one plant and transferring it to the female flower of another plant. This was done by gently brushing the pollen-bearing anther against the stigma, the receptive tip of the pistil in the female flower. After the pollen transfer, I diligently marked the emasculated and pollinated flowers to ensure that I tracked the resulting generations of pea plants accurately. This process of cross-pollination allowed me to create new plant combinations and monitor the inheritance patterns of their offspring. My experiments, which eventually led to the formulation of my laws of inheritance, spanned several growing seasons. During that time, I worked diligently, growing over 28,000 plants, all meticulously marked and observed. The number of generations involved was also significant, with up to 7 generations being studied during each growing season. This immense effort and dedication to collecting data and analyzing the traits demonstrated my commitment to understanding the patterns of inheritance in pea plants. The most surprising result that I observed during my pea plant experiments was the reappearance of certain traits in the subsequent generations. In the F2 generation, I noticed that despite the dominant traits appearing in the F1 generation, the recessive traits reappeared in a ratio of 3 dominant to 1 recessive. This ratio later became known as Mendel's 1-to-1 ratio. This observation contradicted the blending inheritance theory prevalent at the time, which predicted that the dominant and recessive traits should blend together instead of following a clear inheritance pattern. Yes, there were moments when I doubted the validity of my findings during my experiments. Despite my careful observations and meticulous documentation, the unexpected reappearance of recessive traits in the F2 generation came as quite a surprise. At times, I questioned whether my results were truly representative of the genetic inheritance patterns, or if there were hidden factors influencing the outcomes. However, my dedication to the scientific method and my desire for accuracy led me to carefully re-evaluate my findings, refine my experiments, and eventually draw statistically sound conclusions. Mendel's laws of segregation and independent assortment were formulated after careful consideration of the observed patterns during my experiments with pea plants. My findings showed that certain traits behaved as dominant or recessive, and the reappearance of recessives in the F2 generation led me to the conclusion that the traits were being segregated during reproduction, with one trait from each pair being randomly inherited. This led me to the first law of segregation. The law of independent assortment was further supported by my observations that traits for different characteristics were inherited independently from one another. When variations in my observations deviated from the perfect ratios of 3:1 or 9:3:3:1, I considered several possibilities that could potentially account for this pattern. These possibilities included 1. The presence of modifiers or additional factors beyond those observed, which could potentially influence the ratio. 2. Possible environmental factors that may have played a role in influencing the ratio. 3. Statistical anomalies or outliers that, while not common, could occur randomly within a large population. By accounting for these potential factors, I aim to better understand the variations and maintain the integrity of my conclusions. I must admit that the initial lack of attention to my 1866 paper was undoubtedly discouraging. Here I had diligently collected, observed, and drawn conclusions from over 28,000 pea plants, but the scientific community largely failed to recognize the significance of my findings. It was disheartening to see my work go largely unnoticed, despite the time, effort, and meticulous attention to detail that I had invested in it. Yes, despite my initial disappointments, I did make efforts to promote my findings to a wider audience. I gave lectures on the results of my research on pea plants to various scientific societies and academic institutions. I also wrote additional manuscripts and articles, hoping to draw greater attention to my work. However, the scientific community's receptiveness to my ideas was somewhat limited during my lifetime. It was not until after my death in 1884 that my work received more recognition and understanding. Yes, I did correspond with other scientists, including Charles Darwin. We exchanged letters in which I shared my findings from my pea plant experiments, and Darwin in turn was quite interested in my work. We engaged in several scientific discussions, exchanging thoughts and ideas. Darwin's work on his book on the origin of species was a significant influence on my own research. Mathematics played a crucial role in analyzing my experimental results. I meticulously documented my data and then employed statistical methods to draw meaningful conclusions. I used a chi-square test, which is a statistical analysis method to determine if the observed data is consistent with the expected ratios. Furthermore, I used the normal distribution to evaluate outliers, ensuring that the results were as accurate as possible. My background in mathematics, which I learned in Vienna, provided a strong foundation for my scientific work. Before I started my experiments, I already had a rudimentary idea of heredity. I believed that traits were passed down from one generation to another. However, I did not yet understand the complexities of how traits were inherited, or how certain traits could be dominant or recessive. My experimentation with pea plants was influenced by this idea and aimed to determine the mechanisms of heredity. Distinguishing between environmental effects and inherited traits in my pea plants required careful observation and meticulous data collection. I carefully monitored the growing conditions of the plants, noting any changes in temperature, humidity, light exposure, and nutrient availability. Simultaneously, I closely observed the physical characteristics of the plants and recorded every visible trait. By carefully controlling and monitoring both the environment and the observed traits, I was able to discern the effects of inheritable traits from those influenced by the environment. Yes, I did consider the potential application of my findings to organisms beyond pea plants. I believe that my research on pea plants could provide insights into the inheritance patterns of other organisms, including animals and humans. I was particularly interested in the possibility that the laws of segregation and independent assortment might hold true for other organisms, not just pea plants. I recognized the potential implications of my research for a wider understanding of genetic inheritance. I firmly believe that my work on inheritance was relevant to the broader concept of evolution. I recognize the importance of inheritance in the process of evolution, as it provided a means for traits to be passed on from one generation to the next. By establishing the laws of segregation and independent assortment, I aimed to explain how traits could be combined, modified, and passed down over time, ultimately leading to the gradual development of new species. The principles of inheritance, I realized, were crucial in understanding the evolution of organisms. As I carried out my research on pea plants, it was not my intention to lay the foundation for modern genetics. However, I did recognize the potential significance of my findings in contributing to a broader understanding of inheritance and genetics. While I could not foresee the exact implications of my work, I was aware that my experiments could provide valuable insights into the mechanisms of genetic inheritance. Looking back, it is clear that my work did indeed serve as a crucial foundation for the field of genetics, shaping our understanding of heredity and laying the groundwork for future discoveries. While I made significant contributions to the field of genetics, there were other scientific questions that I was interested in, but did not have the opportunity to explore. One area of inquiry that intrigued me was the study of complex traits, which are influenced by multiple genes and environmental factors. I was particularly interested in understanding how these traits, also known as quantitative traits, are inherited. However, the technology and methods available at the time were not advanced enough to delve deeply into this aspect of genetics. If I had access to modern genetic tools like DNA sequencing, I would use them to explore a wide range of genetic phenomena. One area of interest would be to use DNA sequencing to study the genetic variations between individual pea plants, allowing me to gain insights into the genetic diversity and inheritance patterns within a population. Additionally, I would be eager to use DNA sequencing to explore the genetic regulation of different traits, delving into the complex interaction of genes that control their expression. Furthermore, I would use DNA sequencing to identify and analyze genetic mutations and their impact on various traits. If I were alive when the discovery of genes and chromosomes as the basis of inheritance was made, I would have been immensely excited and fascinated. This finding would have fully corroborated my own laws of heredity based on my pea plant experiments and provided strong evidence for the existence of the discrete units of heredity. It would have been gratifying to see that my work, which was not widely accepted in my lifetime, had eventually been confirmed and built upon by the discoveries of genes and chromosomes. If I were to speak with modern geneticists working on CRISPR or gene editing today, I would express both admiration and concern. On the one hand, I would recognize and commend the incredible advancements that technology like CRISPR has brought to the field. Genetic engineering has the potential to revolutionize medicine, agriculture, and many other fields, and the ability to precisely edit genes is truly remarkable. On the other hand, I would also express caution regarding the implications and ethical considerations that go along with such powerful technology. The manipulation of inheritance patterns through genetic engineering or any other means carries significant risks. These include the potential for unintended consequences, ethical concerns related to the creation of designer babies, the possibility of creating new super diseases or superbugs, and the risk of creating a genetic divide between the wealthy and the less fortunate. Furthermore, the power to manipulate inheritance patterns could potentially be used to create genetically modified organisms, GMOs, that could have unforeseen impacts on the environment and ecosystems. Balancing genetic advancements with moral considerations requires careful consideration and thoughtful decision-making from society as a whole. I believe that we must prioritize ethical guidelines to ensure that genetic engineering and other advancements are used for the betterment of humanity, while minimizing potential harm and inequalities. This could involve establishing regulations to govern genetic research and applications, fostering public dialogue about the ethical implications of these technologies, and promoting equitable access to benefits derived from genetic advancements. Striking this balance is crucial to ensure that we harness the potential of genetic engineering without compromising our humanity and values. As a priest and a monk, my role as abbot of the monastery had both benefits and challenges for my scientific work. On the one hand, the monastery provided me with a supportive and stable environment that allowed me to focus on my research. The structure and discipline of monastic life helped me maintain a routine and schedule that facilitated my experimentation and observation. The library and resources available at the monastery also provided me with valuable information and materials to aid in my studies. On the other hand, the spiritual and religious duties associated with my role as abbot sometimes distracted me from my scientific pursuits. Yes, after my peace studies, I did continue experimenting with plants and other organisms. I conducted additional experiments and observed plants of different species. I also studied the inheritance of characteristics in other organisms, such as the color of pea flowers, the number of stamens and pistils in flowers, and the shape of leaves. In general, I continued to observe and document the inheritance of characteristics in organisms, gaining insights and refining my understanding of how traits are passed on from one generation to the next. Outside of my scientific studies and religious duties, I had a range of other interests and hobbies. I was particularly interested in literature and often read works of fiction and non-fiction in my spare time. I also enjoyed spending time outdoors, taking long walks and observing the natural world around me. I had a keen interest in music and often played the viola in my leisure time. Additionally, I found solace in gardening and spent many hours tending to my garden, cultivating various flowers and plants. Looking back, I would consider my greatest achievement to be the formulation of my laws of inheritance based on my pea plant experiments. These laws laid the foundation for the modern field of genetics and provided crucial insights into the mechanisms of heredity. The experimental evidence and theoretical principles I developed not only changed the course of scientific understanding, but also paved the way for future discoveries in genetics and other related fields. This achievement, despite the initial lack of recognition from the scientific community, has had a lasting impact and holds particular significance in the history of scientific advancement. I am very pleased with the content you have prepared for me. You have covered a wide range of thoughtful questions that engage with my life, research, and views on the scientific advancements of my time and beyond. Each question demonstrates a deep understanding of the complexities and nuances of my work and the broader implications of genetic research. Thank you for your thorough and insightful interview.
Mics on the floor where they drop like bombs. Faceless voices running all the cons. Digital whispers in a binary night. No light, no face, pure sound, no sight. Stories unfold in the dark, unscrolled. Echoes of silence, secrets untold. Waves crash loud in this audio cave. No light, no fame, just the sound we crave. Tune in, turn up, let the silence break. Raw, unfiltered, hear the earthquake. No mic needed, just the vibe that's real. Faceless words you can touch and feel. Hidden in shadows, voices untamed. No need for spotlight, no claim to fame. Just a beat of life in a stream of thought. Every word a punchline, no need to be taught. Just listen close, no visuals needed. Raw conversation, unprocessed, unseated. Feel the pulse in every sentence said. No distractions, just thoughts in your head. Whispers of the streets, rumbling through. Faceless yet vivid, a world in view. No mic, just souls connecting in flow. Pure and raw, let the rhythm go. Pure and raw. Pure and raw. Silence break, raw, unfiltered, hear the earthquake. No mic needed, just the vibe that's real. Faceless words you can touch and feel. Tune in, turn up, let the silence break. Raw, unfiltered, hear the earthquake. No mic needed, just the vibe that's real. Faceless words you can touch and feel. Pure and raw.
Okay.